So the central dogma of biology is replication to transcription to translation, which is the process of turning a DNA into a protein. So it starts with DNA replication, which occurs in the nucleus. So it's the process of producing two identical DNA daughter strands from a parent strand. In DNA replication, it's a semi-conservative process. That means that two parent strands split and act as a template strand for the production of the new complementary strand. So the new strand is always one parent and one daughter cell or strand. The process has two stages: initiation and elongation. So initiation begins with the initiation protein DNA-A, which binds to the origin of replication. Then a helicase comes in and unwinds the DNA by breaking the hydrogen bonds between nucleotides. So this causes torque of DNA when it starts cutting. So DNA gyrase or a topoisomerase reduces torque of the DNA strand. Also, single-stranded binding proteins then bind to each template strand to keep the DNA helix open. So that's initiation. Then it goes into elongation. Elongation is when the DNA polymerase attaches to each strand and catalyzes the bond formation between adjacent nucleotides in the 5' to 3' direction for both strands. Because they're both 5' to 3', there's a lagging strand and a leading strand. The lagging strand is a discontinuous strand made of Okazaki fragments. The leading strand is a continuous strand. These Okazaki fragments are short, discontinuous fragments on the lagging strand, and they're eventually joined together by DNA ligase to make a continuous strand. So that goes into the second part, which is transcription, which also occurs in the nucleus. Transcription is the process of turning those DNA strands into RNA. So the genes on DNA code for many different RNAs. But mRNA is the only RNA translated and is used for the coding of proteins. The DNA is also translated into rRNA, tRNA, different RNAs like that, but mRNA is the only RNA translated. So there's 3 steps here. It's initiation, elongation, and termination. So it starts with the initiation. In initiation, RNA polymerase bonds to the promoter sequence on the gene, which is the promoter is the Tata box, which is 10 nucleotides upstream. Also, then sigma factors binds to the RNA polymerase to make it a holoenzyme. This region of DNA is unwound and phosphodiester bonds form between the first 2 nucleotides. Then the sigma factor leaves. Now elongation starts. Once the sigma factor leaves, RNA polymerase begins to move along the template strand in the 3' to 5' direction on the template strand. RNA polymerase elongation creates an mRNA strand. So then you have termination, which is the end of transcription. So there's 2 types. There is extrinsic, which is row dependent. So row factor binding site happens, and it is adenine rich, that site. Then there's the intrinsic, which is row independent, which a hairpin loop forms and causes a hairpin loop forms at a cytosine rich site. Intramolecular hydrogen bonds causes the loop to form, and eventually the strand breaks, making the end of the mRNA and the end of transcription. This leads into translation. Oh, actually, you had post-transcriptional modifications first. So mRNA gets modified by poly-A tails, guanine caps, a 5' guanine cap, a 3' poly-A tail, and splicing of the introns. So these help make a mature mRNA, which can then be used to code for the protein in translation. Translation is the making of proteins from RNA. There's 3 steps: initiation, elongation, and termination. Initiation starts with the sequence of AUG at the 5' end of the mRNA. At this point, the small ribosomal subunit comes in and binds to the mRNA. In prokaryotes, the small ribosomal subunit first binds to the Shine-Dalgarno box and then to the AUG sequence. Then the large ribosomal subunit comes and binds to the small one. Large ribosomal subunit is where peptidyltransferase activity occurs. This holds 3 tRNA binding sites of the exit, which is E, the amino acyl, which is the A, and the peptidyl site, which is the P. Then it goes into elongation. In elongation, the first sequence of the AUG codes for methionine in eukaryotes and formulated methionine in prokaryotes. The methionine tRNAs are positioned in the P site, and this is the only time this happens. Then a new and charged tRNA comes and binds in the A site with help of elongation factors. So then new tRNA carries an amino acid coded for by the mRNA strand. Then peptide bonds form between amino acids on tRNA in the A site and amino acids of tRNA in the P site. The growing polypeptide chain is transferred to tRNA in the A site. Translocation now occurs once the growing polypeptide chain forms, and the empty tRNA moves into the E site and is discharged from this ribosome, and the tRNA with the growing polypeptide, which was in the A site, moves to the P site, and the P site is the place where the growing polypeptide chain with the tRNA always is. This leaves the A site empty for the next charged tRNA to bind to. This leads to termination. This happens when the A site of ribosome comes upon a stop codon. The 3 stop codons are UAA, UGA, and UAG. No tRNA carries an anticodon for these 3 codons, so release factors bind to the stop codons, then release of the ribosomal subunits, mRNA, and newly formed polypeptide are released, which that is the new protein.